Welcome to this lecture on reproductive biology. Today, we are going to explore two of the most critical and perfectly synchronized events in human reproduction: oocyte maturation and ovulation. Imagine a marathon runner, frozen in place, just waiting for the signal to sprint to the finish line. That is exactly what happens to a human egg cell. Let us dive in and understand how this incredible process works. To begin, we must understand that an egg cell, or oocyte, does not develop all at once. It actually begins its development before a woman is even born, but then it hits a biological pause button. It stays arrested in a stage called prophase 1, specifically the diplotin stage. During this long sleep, it contains a large nucleus, known as the germinal vesicle. It will remain in this frozen state for years or even decades, waiting for a massive hormonal signal to wake it up. Now that we know the egg is asleep, what wakes it up? The brain releases a massive spike of luteinizing hormone, or LH, which acts as the ultimate biological alarm clock. This surge causes the germinal vesicle to break down. The cell then resumes meiosis, dividing its genetic material in half, so the resulting embryo will have the correct number of chromosomes. The unneeded chromosomes are pushed into a tiny waste packet called the first polar body. Finally, the oocyte pauses one last time at metaphase 2, fully mature and ready for a sperm. This biological alarm clock relies on precise chemical signaling. To understand how the LH surge triggers maturation, we look at the Michaelis-Menten binding equation. Conceptually, this formula describes how hormone molecules floating in the blood bind to receptors on the cells surrounding the oocyte. B represents the number of activated receptors. B max is the total number of available receptors. The concentration of LH is in the brackets, and KD is the dissociation constant. As the hormone concentration increases, more receptors are activated, eventually leveling off when all receptors are full. Before we move on, let us see this with a concrete example. We want to calculate the number of activated receptors during the normal resting phase of the menstrual cycle. We are given a maximum receptor count of 10,000, a dissociation constant of 15, and a baseline LH concentration of 5 units. We substitute these values into our binding equation. The denominator becomes 20. Dividing 5 by 20 gives us 0.25, or 25%. Multiplying this by 10,000 gives a final answer of 2,500 active receptors. This low level keeps the egg peacefully asleep. So we have seen the resting state. This brings us to proving how the surge acts as a biological switch. We now evaluate the situation during the massive LH surge, where the concentration spikes to 85 units. We substitute 85 into the equation. The denominator now becomes 100, reflecting the massive influx of hormones. The math reveals that 85% of the receptors are now bound, resulting in 8,500 activated receptors. This sudden, overwhelming flood of signals crosses the critical threshold, proving mathematically why the oocyte immediately resumes meiosis. Now that we have proven the mathematics of the trigger, let us look at the physical transformation. On the left, you see the dormant oocyte in the germinal vesicle stage, with its large, intact nucleus. Following the LH surge, represented by the arrows, we move to the middle stage, germinal vesicle breakdown. Here, the nucleus dissolves. Finally, on the right, the cell reaches metaphase 2. Notice the small structure at the top right of the cell. That is the first polar body, holding the discarded half of the chromosomes. The egg is now mature. With the egg now mature, it needs to escape the ovary. This brings us to the mechanics of ovulation. Think about blowing up a thick rubber balloon. If you just add water, it takes massive pressure to pop it violently. But if you place a tiny drop of acid on one spot, it weakens and bursts gently. This is exactly how ovulation works. The follicle does not explode from high pressure. Instead, following the LH surge, surrounding cells secrete powerful enzymes like collagenases. These act like microscopic scissors, degrading the collagen fibers that give the wall its strength. As these enzymes do their work, the follicle wall becomes progressively thinner at one specific point, which we call the stigma. Eventually, the wall can no longer contain the fluid inside, and it gently ruptures. The follicular fluid carries the mature egg out safely. This precise, enzyme-driven mechanism is crucial. If the body relied on massive pressure, a violent explosion could severely damage the delicate egg or the ovarian tissue. Understanding this is also vital for treating infertility, as a lack of these enzymes leaves the egg trapped inside the ovary. To truly understand how this gentle rupture happens, we can look at the physics of spherical vessels using Laplace's law. Conceptually, this formula explains how the tension in the wall of a sphere relates to the pressure inside, its size, and its thickness. T represents the wall tension, which is the force trying to tear the tissue apart. P is the outward fluid pressure. R is the radius of the follicle, and W is the thickness of the wall. Notice that if the wall thickness W decreases, the tension T will increase dramatically. Let us calculate the forces at play to see exactly why the follicle ruptures. We will start by finding the baseline tension before the LH surge. We are given an internal fluid pressure of 20 millimeters of mercury, a follicle radius of 10 millimeters, and a healthy thick wall of 2 millimeters. We substitute these into Laplace's equation. The numerator becomes 200, representing the total outward force. The denominator becomes 4. Dividing 200 by 4 gives a final answer of 50 units of tension. This is a stable, manageable stress, so the follicle remains completely intact. Now that we have our baseline, we will mathematically prove that enzymatic thinning is the primary driver of rupture. 12 hours after the LH surge, digestive enzymes have severely thinned the wall at the stigma down to 0.5 millimeters. The pressure and radius remain exactly the same. We substitute the new wall thickness into the denominator, which now becomes 1. 200 divided by 1 gives 200 units of tension. The tension tearing at the tissue has quadrupled simply because the wall became thinner. This massive spike exceeds the tissue's mechanical strength, causing ovulation. Let us visualize the physics we just calculated. In this diagram, you see the large spherical follicle. The outward arrows, labeled P, represent the constant internal fluid pressure. Now, look at the magnified inset on the right. It shows the wall thickness, W, and the stretching tension, T, pulling the tissue apart. Because the enzyme specifically degraded the wall at the top, creating the thin stigma, the tension there skyrocketed. This localized mechanical failure allows the oocyte, surrounded by its protective cumulus cells, to gently squeeze out of the ovary without a violent explosion. We have covered a lot of ground today, so let us summarize the key takeaways. First, Oocyte maturation is essential to have the genetic material, ensuring the future embryo has the correct number of chromosomes. Second, the LH surge is the master biological switch that triggers this awakening. Third, ovulation is a gentle, enzyme-driven process that thins the follicle wall, rather than a violent pressure explosion. Finally, as Laplace's law proves, thinning the wall drastically increases tension until rupture occurs. Together, these perfectly synchronized events transform a dormant cell into a new life. Great job following along.